tonight by the Honorable Rector of the University of Urbino, Carlo Bo, Giorgio Calcagnini, and Professor Giovanni Bottegoni. The Honorable Rector, Giorgio Calcagnini, will now speak.
Good morning, everyone, and I'm very pleased to have you here for this important recognition of Professor Changeux. I'll read the reasons behind the fact that we honored Professor Changeux with the seal of the University of Urbino. Professor Jean-Pierre Changeux is internationally renowned for his outstanding scientific accomplishments and leadership in the field of neuroscience, particularly for his pioneering contribution to the study of and understanding of neuroreceptors over the past 5 decades. Having pursued his PhD studies at the Pasteur Institute in France under the guidance of Jacques Monod and François Jacob, Professor Changeux obtained his doctorate in 1964. He then embarked on postdoctoral studies, first at the University of California, Berkeley, and later at Columbia University College of Physicians and Surgeons, New York. Returning to France, he served as an assistant to the chair of molecular biology held by Jacques Monod. In 1972, he assumed the directorship of the Unit of Molecular Neurobiology at the Pasteur Institute. Moreover, he was concurrently elected professor at the Collège de France, holding the chair of cell communications until 2006. Presently, Professor Jean-Pierre Changeux holds the position of Emeritus Professor of Neuroscience at the Institut Pasteur and is Chair of Cellular Communications at the Collège de France. He also serves as Honorary President of the Neuroscience Department at the Institute of Pasteur. Since 2006, he has been an international faculty member at the Kavli Institute for Brain and Mind, University of California, San Diego. During his doctoral studies, Professor Changeux investigated the allosteric regulation of enzymes, which culminated in the development of the concerted transition model for allosteric proteins, later known as the MWC model. This conceptual framework remains pivotal in explaining the function of cooperative proteins. In his thesis, Professor Changeux proposed that signal recognition and transmission across membranes, particularly at synapses, might employ mechanisms akin to the allosteric regulation of enzymes. This hypothesis initiated over 40 years of groundbreaking research dedicated to unraveling the structure of a specific receptor and its function. His work has provided profound insights into brain chemistry and brain-mind relationship. The publication of his seminal book, Neuronal Man, The Biology of the Mind, in 1985, brought Professor Changeux recognition beyond academia. Subsequently, he authored and co-authored several other books inspired by his teachings at the Collège de France, and he's curated 3 major exhibitions on art and science. In acknowledgement of his contribution, Professor Jean-Pierre Changeux has received numerous prestigious prizes and awards, including the Albert Einstein World Award of Science in 2018, Goldman Rakic Prize in the same year, Olav Thorn International Research Award in Biomedicine in 2016, National Academy of Science Award in the Neuroscience in 2007, Balzan Prize for Cognitive Neurosciences in 2001, Linus Pauling Medal in 1998 and 1999, Louis Gente Prize for Medicine in 1993, Wolf Foundation Prize in Medicine in 1983, Richard Lounsbury Prize in 1983, Gardner Foundation International Award in 1978, among others. In 2005, he was honored with the Lewis Thomas Prize for writing about science by Rockefeller University, New York. Professor Changeux has received honorary degrees from universities worldwide, including the University of Turin, École Polytechnique Fédérale of Lausanne, University of Southern California, Los Angeles, the Hebrew University of Jerusalem, Ohio State University, and the University of Buenos Aires. He was a member of foreign members of numerous prestigious scientific and literary academies globally. In 2010, he received the Grand Croix dans l'Ordre de la Légion d'honneur. In recognition of his unparalleled achievements, the University of Urbino is honored to bestow his highest honor upon Professor Jean-Pierre Changeux, the Great Seal of the Università degli Studi di Urbino Carlo Bo.
Thank you, Giovanni, for your very distinguished honor of receiving the seal of the University of Urbino. And I wish to say that for me, it is a symbol, a seal between the research being done in your university and the Collège de France, and also a seal between the many Italian scientists who work in my laboratory. And I would like to mention a few of them. And first, Giacomo Giacobini from Torino on the role of nicotinic receptor in the embryonic development. Also Cesare Montecucco in Padova, who work on the action of toxin on receptors. I mentioned Maurizio Brunori, who work on hemoglobin and allostery, who is an old friend for many, many years. And last but not least, Marco Cecchini, who is here. And I will not mention now what he has been doing, but during my presentation, because he has contributed a lot to what I will discuss in the second part of my presentation. So I will be... I should keep my hat or not? Yeah.
As you like, as you prefer.
Maybe I take it out. Yes, thank you very much. It's the first time that I give a talk on Allostery with such a dress, but it's an exceptional event. And so I would like to just say a few words about ALLODD, which has been founded by Curie on European scale for the study of allosteric protein and drug design. And I'm especially pleased today to be in Italy with Professor Giovanni Bottegoni, Roman Zubarev, and Ijen Chen, who are the chairs of this meeting, and for the invitation to give what you call Lectio Magistralis, in addition to this beautiful seal that I received now. So the theme of my presentation will be allosteric modulation of neurotransmitter receptors toward a new pharmacology. And just before going into the essence of my presentation, I would just to show you on the left of my side a document from Katrina Munt about the Broca region in the brain, and all the different colors show different receptors. So there is a considerable diversity of receptors in the human brain, and it is, of course, of great importance to try to understand, as we shall see, brain communication in this presentation. So first I will perhaps go back about 50 years ago. I'm sorry for those who already heard me there, but I think sometimes it's good to go to definition because sometimes they are not understood as they are. So I will talk about Allostery. So where did this concept come from? I think we were in the 50s, 60s, and at that time Norbert Wiener had introduced cybernetics. And in his presentation of cybernetics, I had the pleasure to attend one of his presentations. The notion of feedback was, of course, mentioned, and being a critical mechanism working on these cybernetic machine or on our models. So at that time, I got interested into the mechanism of such feedback inhibition, and Monod and Jacob mentioned to me the recent discovery by Erwin Umberger that in sequences of chemical reaction going from threonine to isoleucine in bacteria, the end product of the pathway, which is isoleucine, is in fact inhibiting the first enzyme of the pathway. So there was a specific feedback inhibition of threonine deaminase by isoleucine, and there was a threshold also involved in this effect. Now, I, of course, became interested by the mechanism of this type of feedback inhibition. And very early on, I had in mind that there were a specialized kind of mechanism involved in feedback inhibition, and try to dissociate feedback inhibition from catalytic activity. And I found that some heating of the enzyme led to an uncoupling, as you can see here, of the effect of isoleucine on the enzymatic activity, which stayed when the feedback inhibition is lost. Similar results were found with aspartate transcarbamylase in America, and Jacques Monod, when I made this finding asked me to make a presentation at the Cold Spring Harbor meeting in 1961. If I mention it, is that because this opportunity, I showed that two models could account for the feedback inhibition, overlapping sites, steric hindrance, classical competitive interaction, or a model where two topographically distinct sites were involved, one for the substrate of the enzyme and one for the feedback inhibitor. So the proposal was made that this feedback inhibition was mediated by distinct sites through, of course, the change of the protein. And throughout my thesis work, I developed the issue and in particular studied the cooperative interaction which are mediated by threonine. I mentioned the threshold effect. And then the idea came that, in fact, we had some kind of cooperative organization of the protein, here a dimer, but could be more, as you shall see, and then with axis of symmetry, and that there would be a spontaneous transition between resting and active state, which would be some kind of cooperative interaction between, let's say, a resting inactive and an active state. So this proposal was suggested for several reasons I am not going to discuss here. But the idea that there are pre-existing conformation and isomerization constant L to qualify this equilibrium was something that was difficult to accept by many colleagues who prefer the induced fit à la Cochonde. And they say, why are you complicating our life, and so on and so forth. So this is nevertheless, and again, there were several reasons for us to propose that with Monod and Wyman. And first of all, we were able to formalize the equation for binding and for conformation. So what we call state function and binding function. And of course, one of the critical features of this model was that the state and binding function should not superimpose. If there was an induced fit, then one could have the conformational function just being closely following the binding function. So this was a critical aspect to test for this Darwinian model opposed to the Lamarckian view of Cochonde, if I may say. And the work was done with John Gerhardt and Howard Scheitman, one as was postdoc in their lab. And here you can clearly see that in the case of aspartate transcarbamylase, this is one example among many others. The conformation function or the state function does not become superimposed with the binding function. On the opposite, they are strikingly different, and this was also confirmed by Lipscomb and collaborators with the crystalline structure of this enzyme. Quantitative fit by the equation was obtained, and this was, if I may say, validation, the first validation of the two-state model with ATCs, which was nevertheless criticized and is still criticized by some of people who really want to defend the induced fit, which in fact we can include in the general model. So there is, to my opinion, not opposition, but some way of including it in our more general model. So then the question raised in my thesis work that possibly this kind of allosteric transition could play a role in brain communications. And this is why I became involved in the identification of the nicotinic receptor. Because at that time, there was no receptor for neurotransmitter being identified. So that was really the first one. And I started as a further postdoc with David Nartmanson, who had developed the fish electric organ as a source of synaptic material. And I had the opportunity to meet Chen for Taiwan, who had been throughout his life working with venom from snakes from Taiwan, and discovered that one toxin was extremely specific for the postsynaptic receptor. And therefore, I mixed the two, a fish and a snake, and it came out that protein could be identified, purified, and this gives the first EM structure of the receptor, both purified on the left from Electrophorus, which is also an electric fish from the Amazon. And on the right, we had the opportunity to look at the postsynaptic membrane, purified postsynaptic membrane. And as you can see, two things. First, there are perhaps pentamers, and also, very importantly, that the density of receptor on the postsynaptic membrane is very high. This was for Jean Carteau when he was a student with me and Marco Benedetti. So this led to further studies by, in particular, Carlin, Raftery, and Huchot in my lab. And the structure, which was essentially characterized by Carlin, was 2 alpha, 1 beta, 1 gamma, 1 delta, an heteropentamer. So this was quite a problem for me, in particular, not for them, but for me, because I was, of course, thinking of a symmetrical protein. And it looked as if there were no symmetry. Then we identified the first 20 amino acid of the sequence of the alpha subunit. Then subsequently, after he and others identified the sequence for the beta gamma delta subunit, and they found that there were structural homologies between these different subunits, and therefore that symmetry was not completely lost. On the opposite. So at that time, the group of NUMA, of Heinemann and ourselves, get involved in identifying the full amino acid sequence, and we distinguished in this full sequence two domains, an extrasymmetric domain and a transmembrane domain, and that was the first two-dimensional model for the subunit of the nicotine receptor. And of course, the temptation was that the site for acetylcholine was in the extracellular domain and the ion channel in the transmembrane domain. So this had to be subsequently demonstrated, and this was done in our lab, largely, and other labs as well. But first of all, the identification of the acetylcholine binding site. There are two interesting characteristics there. One, it is that it has the boundary between subunits. I will come back on this issue, which is a very important point for the linking the primary and quaternary structure of the receptor. And also, as you see here, that there are a lot of aromatic amino acids, and therefore this is some kind of aromatic pocket. And this was found, of course, in the synaptic domain of the sequence. So the other thing was the ion channel, and we proposed that it was located in the transmembrane domain 2 through affinity labeling reagents with a compound called chlorpromazine. Remember this hydrophobic amino acid within the ion channel. I will come back on that. So the conclusion was that acetylcholine binding site and ion channel are structurally distinct. And then the question was the relationship between the two. The distance between the acetylcholine binding site and the ion channel was estimated by fluorescence spectroscopy by the group of Palmer-Taylor. And here you have an image that I made from the picture of Nigel and Wynne, which clearly show that there are two categories of allosteric interaction taking place between identical sites and between the identical site and the ion channel. So, conclusion, we have a bona fide allosteric protein. Now, for years, we tried to crystallize the receptor and have the three-dimensional structure at the atomic level. And one day I was a referee of a paper by Tasnim and colleague who mentioned that working purely, it's a pure sequence work in bacteria, there were some sequences looking a little bit like the nicotinic receptor. So when I read this, I jumped on my seat, if I may, and asked Pierre-Jean Changeux immediately, after having accepted the paper, of course, first to work on bacterial receptors, which are shown here with Glick, and also the group of Dutzler with Elic. So this was the first atomic structure of pentameric elongated ion channel. And you can see already that between the two, there is a very close similarity. Even the bacteria are very different. And the other aspects of this slide is that following this initial discovery, the group of Guo and essentially Ryan Hibbs and many others have identified the receptor for glucl in worms, GABA, glycine, 5-HT3, nicotinic receptor, alpha-4 beta-2, and even alpha-7. So clearly, we have a highly similar 3D structure of the member of the nicotinic receptor family, and this shows that our brain receptors originate from bacteria. We have to be very humble with our brain. But this is an interesting finding that the structure from the evolutionary point of view have been so well conserved through billions of years. Now, I will very briefly talk about something which is centered to the drug design, which is allosteric modulation. The idea, which was inspired initially from the work of Steinbach on benzodiazepine and GABA receptor, but we developed in the case of the nicotinic receptor essentially with ivermectin and calcium, was that ligand binding to sites which are not the neurotransmitter site and not the ion channel, but other type of sites, could modulate the transition, allosteric transition of the receptor. So here I showed a calcium site. I saw also alcohol, and one of the most interesting one was ivermectin. I show that in a minute. And it means that there is a new category of drugs, which we refer to as allosteric drugs, which are efficient on receptors, but have the privilege of not being identical to the functional site. So it means that there is less interference with brain signaling taking place at their level, and therefore one can develop a new strategy for drug design. Now this is just to show you some of the results with ivermectin. As you know, it is a very potent anti-helminthic acting on glucl. I just showed the structure before, and which was identified by Omura and Campbell. And very early on, in 1998, we discovered we discovered with Daniel Bertrand that ivermectin is extremely strong potentiating agent. You see the change of affinity from normal to activated by ivermectin, and that this is lost by mutation in the transmembrane domain segment. Now, the structure of the site has been identified by Go precisely on GluCl, and you can see that it's beautifully located in the transmembrane segment. So we have a really new category of modulatory sites present in the transmembrane domain. Now, this is, of course, a statement which can be generalized, and I want to mention the work of Marco on a database of glycine receptor allosteric ligand. As you can see, one can distinguish agonists, antagonists, positive allosteric modulator, negative allosteric modulator, and these compounds, interestingly, some of them bind to rather different sites. And this is, I think, a beautiful illustration where you can see that on the subunits of the glycine receptor, there are 6 categories of allosteric sites. And I think this afternoon, Marco is going to talk about one of them here. So this clearly shows that we have a new allosteric pharmacology going on. I will come back. Now, to link all these sites, not only the agonist binding site and the ion channel, but all these allosteric sites which make some kind of constellation on the receptor, we need a conformation change. And there is therefore a need for an allosteric transition to couple these different sites together. And the first evidence for that was a work which was done in my lab in 1979 by Thierry Edman, following the binding of fluorescent agonists, here danacylcholine, on the receptor, and the fact that, in fact, one can follow the conformation transition of the nicotinic receptor by fluorescent spectroscopy and distinguish already several relaxation times, fast, intermediate, and slow. So at that time, we proposed, in fact, to account for the data, that there will be not only an active conformation here, but also some intermediate and desensitized conformation, the agreement with the work of Katz and Teslev. So just from this analysis, it was shown that there was a low intermediate which correspond to the active state, and 2 other states, and in particular, we'll come back very briefly at the end on that, and a desensitized state with high affinity and a closed channel. So this means that we are not only pleased with the mono-Wyman-Changeux model, but they are, which is a 2-state model, but it's a more several-state model. Now, just to illustrate this interesting modulation, I want to show that mutation can have an effect which to some extent resembles the effect of modulatory ligand. And you have here experiment which was done with Daniel Bertrand, which shows that some of the mutation in the channel domain, remember the hydrophobic amino acid, is able to change and shift the affinity of the receptor to our high affinity state. And therefore, we have a constitutive mutation. And of course, some of them are have pathology, are disease. This is the case of frontal lobe dominant epilepsy and also of congenital myasthenia in the muscle. So the receptor from the brain and from the muscle can be mutated to give this kind of phenotype and disease. And the interesting thing I want to mention, the work with Martin Karplus, is that these mutations are located at interface between subunits. And this again links again the site, the amino acid involved in the binding between the subunits and the quaternary change mediating the allosteric transition. So I was, of course, interested by the identification of this transition. I think most of you know the work by X-ray structure with Glick and by molecular dynamics by Marco. You see a nice, molecular dynamic simulation. And we distinguished at that time two movements, twist and bloom, already, both by molecular dynamic simulation and by the X-ray structure. So there is, if one may say at atomic resolution, a two-state switch, which is taking place between the resting and open conformation. So this is of interest because I mentioned that we are dealing with a receptor which is originated from bacteria, and the time scale of this conformational change impose a constraint on our cognitive function. So I talked with physicists telling them, our brain is slow. Your brain is slow, not mine. But that's what I meant. I meant it's slow compared to the computers that they have been using, which work at the speed of light. So this is something which have an important consequence on the function of our brain. So this is the last view we have, and this is in a paper with Marco and Pierre-Jean Changeux in Annual Review of Biochemistry, where the conformational change taking place during activation are elaborated in a more detailed manner. Some loop from the active cycle C loop are moving. Then there is a global tilting and leading to a twist and the opening of the channel. So these are some recent views, in particular here the structure of Yves Nury and of course the molecular dynamic simulation by Marco. So we have now a sophisticated twist and bloom conformational wave for nicotinic receptor gating. And I just want to show, to close, there are still a few slides, not many. So I will be on time, I hope, on the way the conformation change propagates. So the issue is you could have this bloom and twist, boom, like that. Or you could have a progressive twist, let's say from top where there is acetylcholine to bottom. And that was, of course, an interesting possibility, which was in fact suggested by the identification of some intermediate states between resting and open. Now, the method that was used by Pierre-Jean Changeux is what is called fluorometry or fluorescent tryptophan quenching, whatever you can call it, which measure the change of structure and using inner sites where the receptor is expressed, the patch clamp to follow the opening of the channel. And you can see here that there is a shift in time between the fluorescence, which appears sooner than the opening of the channel. And from this critical result, which I think is a very important one. One could derive a model where there is a progressive shift of the conformation change from the top to bottom or from bottom to top. And there is, therefore, some change to further analyze, which is ongoing on that. Now, I will close on some very recent experiments which have been just published in Cell 2004. And this is important for us from a pharmacological point of view. So I mentioned the positive allosteric modulator. And there are two, in the case of the alpha-5 nicotinic receptor, positive allosteric modulator. Ivermectin, I mentioned it already. And you can see there is an increase, a potentiation of the response without any change of the response. And there is another type of receptor, of ligand called PAM2, where there is, in addition, an effect on desensitization. Desensitization is blocked. So the sites are located, of course, in the transmembrane domain for all these ligands. And the question is, how can we account for an effect of some of these compounds just on activation, other on desensitization, or the two together. And this is, of course, a preliminary kind of finding, where, as you can see here, in the presence of PAM1, the channel is closed, and this is a desensitized state, and in the presence of PAM2, the channel is open. So it would mean, as it was in fact predicted by Maria Strikova, working with Marco in the framework of Human Brain Program, that there is indeed the possibility of having both closed and desensitized states. So this is introducing an important concept that there could be a conformation-specific pharmacology, not only a site-specific, not only orthosteric site and allosteric site, but also a pharmacology for each of the individual conformation. And I will close to link the beginning and the end, if I may say. This is the experiment we did in 1978 with Laurent van Dienen. And we showed that there is a very high specificity of interaction of the receptor between cholesterol and long-chain fatty amino acid. And this is the late experiment, 24, by this team, which shows that the lipids are acting in the late stage of the allosteric transition I just illustrated before. And I know that Zoe is also working and may talk about this later on. So this is the end, just to show that in one side, that in fact, this concept I illustrated from strictly basic science, understanding cybernetics of the cell, led to interesting medical application. So I didn't want to go through all of them, of course not, but there are presently 91 allosteric modulators which are used as medicines, and you have here a list of some of them. You recognize the one on GABA receptor and some on PAR receptor, all these are ligand-gated ion channels, but also, of course, GPCR, and therefore we think that many diseases, you can see on the left, can be treated by some allosteric medicine. including the conformation-specific monoclonal antibodies, which are widely used in carcinogenesis. Okay. Validity of the concepts, I think you are, I hope, convinced. Validity of the two-state model, well, more or less, because there are more than two states. Importance of allosteric modulation and conformation-specific drugs, which follow the identification of allosteric transition at atomic resolution, a new allosteric pharmacology. So these are those who did the work, the collaboration with Strasbourg and Institut Pasteur. I already mentioned the name of Marco, Pierre-Jean, Maria, and thank you, Giovanni. Thank you very much. Thank you so much.
Ladies and gentlemen, this Lectio Magistralis concludes the ceremony for the awarding of the University Seal to Professor Jean-Pierre Changeux. Thank you for your participation.